quiz on Thursday. The slides.
There is an extra credit also on Thursday, not this Thursday, the 27th. If you ask me again, I say, I don't know.
And then whatever
My memory becomes selective. James, I can pick on you. You're sitting back there, so I can pick on you. My memory becomes selective when you ask me something that I already said.
What is it? After this class?
5 o'clock at p.m. On Thursday the 27th. Is that right? Amanda, I tried to get one more person, but I tried. I don't know. I'm going to try. Amanda Kana, she's second year med student at UC Davis. So she will be here, ask questions, how to get into medical school, or the requirements for pharmacy school, dental school is pretty much the same. Veterinary school, if you're interested. She's going to be here, and then room 219, where the zoology lab is, from 5 to 6 p.m. And then it's 5 points through until end of semester. We have more. I think we have 5 or more speakers until. So this is the first one. I thought I invited her after your first exam, so you have time. So the rest of the speakers, I don't know, it could be right before your exam, we'll see. We'll see. I have 2 other persons, two of them that I have lined up. They're medical doctors, so they should be here sometime in October. They said October, September is not. Usually they go on vacation in September. You and I go on vacation in August, July, where everybody else is vacationing, so we have to stand in lines. They're smart. They go in September, which there are no lines. They're smart. Okay, so let's go. This is the beginning of the material for exam. Please don't ask me. You are in zoology. You are going to become medical doctor, dentist, pharmacist. So I say something once, right? I say something once, and then take note or just put it up here. Don't ask me. I mean, when the extra credit is, I say, I don't know. I forgot. I mean, what is in the quiz? I don't know. I forgot. I treat you differently than non-majors. The non-major biology class I teach, they ask me a question 20 times. I still answer. But you guys, you're going to medical school. Remember that. Yeah, I say something once. Okay, so the definition of, quickly, fertilization, the process. Fertilization is the name of a process. When sperm enters the egg, not all animals do that. When sperm enters the egg, that is called fertilization. Okay, so quickly, if I run out of question at tip of the sperm, there is a sac full of enzymes. It's called acrosomal sac, and then it dissolves the outer layer of the egg and enters the egg. And what happens in case of human? Just the nucleus of the sperm enters the egg and fertilize the egg, but the tail and the mitochondria do not. So all of the mitochondria, you learned that in Biology 1, you have is from your mother. You do not have any mitochondria from your father. Okay, that's in the case of human. So the only thing you have is the half of you is from your father, which the head of the chromosomes are. And that's the process of fertilization. Quickly, I'll talk about it. What happened here is a picture when sperm enters the egg, and then after sperm enters the egg, you have it's called zygote. It's not up there. It's called zygote. Zygote is Z-Y-G-O-T-E. Zygote is fertilized egg. They call it fertilized egg. That is called zygote. And then after you have one cell right here, for example, this is one cell. After one cell, then this undergoes mitosis. You learn in Biology 1, becomes 2 cells. And then 2 cells become 4 cells. Somebody, I guess, took away the 2 cells model, hang it up from the back of the truck or the car. Have you seen that? You know what I'm talking about. Have you seen those? It's missing. My 2 cells is missing. So after 1, I go to 4. So it's 4 cells. After 4 cells, it's 8 cells. Right here, 8 cells. After 8 cells, it's what? 16. Process of mitosis. You learn that from Bio 1. So here is the sperm flying, swimming to the egg, fertilize the egg. That's the name of the process. And then cleavage occurs. When in biology 1 you learn mitosis, but the term that we like to use now is cleavage. Okay, or early embryonic development. Okay, is called cleavage. And then after that you have gastrulation, which I will talk about it here in a minute. And then you have organogenesis, when the organs are being formed. Gastrulation, when you have the germ layers are being formed, and I will talk about it today. And finally you have the growth, and you have a human, or in this case, sea urchin is drawn there. Okay, reproduction, main events, gamete formation by what process? Gamete is formed by what process? Meiosis. Meiosis, not, the T is not in there. And then what happens during meiosis? Meiosis in Greek means reduction. What are you reducing when you make, when your body, when you are doing meiosis? You're what?
Ploidy level. Start haploid cells and then haploid cells.
What is haploid? What is meiosis? When early people discovered this, they said, what name are we going to give it? Meiosis, it means, in Greek, it means reduction. You are reducing the number of chromosomes. Okay, that's what happens. And mitosis, it means thread. The multiplication, subtracting. But anyhow, I'm not going to go over those in biology. Anyhow. Fertilization, after fertilization, you have cleavage. I just talked about that. Cleavage, at the bottom line is mitosis, gastrulation, and then neurulation, formation of the nervous system, so on and so forth, organogenesis, formation of the organs, and finally growth and birth. Embryology, fertilization, zygote, I talked about that. Zygote, it means fertilized egg. When sperm enters the egg, is called zygote. What if there are animals that they undergo parthenogenesis? What if there is no sperm involved? Fish, some fishes, some lizards, some frogs are like that. Just the egg becomes an entire organism. And that is called parthenogenesis. When there is no involvement of sperm. Yes, sir.
Isn't that mitotic?
Amitotic.
Okay.
Amitotic. A in front of the word then, without fertilization. Okay, so it's called parthenogenesis. That's when there is no sperm involved. And then, here we go, I gave you examples. Rotifer, you don't know what rotifers are, but you know what some crustaceans, some insects, some fishes, desert lizards, etc. all there are. And what is cleavage? I tossed the term before, but I did not give you the definition. Here is the definition of cleavage. Cleavage is the process of nucleus and cell division in zygote. And that's pretty much what you have zygote. You know, as I said, when you cut your skin, mitosis is happening here. When you have a little baby, right, growing taller and taller, and if you have a plant, it's growing, that's mitosis. Am I making some sense? This is also the process of 1 cell to 2 cells, mitosis, but since this is happening in the zygote, right, look at the definition. They like to differentiate it, the terminology. They like to differentiate it a little bit from mitosis. They call it cleavage. Okay, so I use the term cleavage more here. Types of cleavage, there are 4 main types of cleavage, and I'll have to go over and I will talk about them, and it's going to be in your quiz and exam. Radial holoblastic, spiral holoblastic, discoidal meroblastic, and rotational holoblastic, which I will talk about them as time comes along. Okay, early cleavage, cleavage and early development, an animal vegetal axis is visible on the embryo before cleavage begins. Here we go, a vegetal pole. We always had that before, but I never, students always do ask me, what is vegetal pole? Formed by the presence of yolk at only one end And then animal pole, where the regions are containing mostly cytoplasm, and then establish polarity in the embryo. So when you look at, in the lab, you don't have to worry about this. Looking at under microscope, see which end of the egg or the after fertilization occur is vegetal pole, and which end is the embryonic pole, animal pole. But it is important when we start talking about different type of cleavage. I hope I'm not getting you lost. When you study these things a little bit more, and then you say, okay, I mean, which end, how do you know it's happening horizontal or vertical? Okay, I will toss the term at you in a minute, in a few seconds, the term horizontal and vertical. The term horizontal and vertical has to do with this, the vegetal and animal pole. Okay, are you guys ready? Let's look at it. Right here, the first one is C star. For example, you have a radial holoblastic. Oh, yeah, and this one, I want to mention the term holoblastic and meroblastic, the terminology. Holoblastic, it means when you have a zygote and the entire cell divides. That's called holo. Holo, it means complete, all. The whole entire thing. That's what that is. When you have cleavage, that happens in the zygote, that happens all around. Then meroblastic, it means you have a thick yolk, a very thick yolk. So the cleavage does not occur, the mitosis does not occur all around of the entire zygote. You have partial cleavage, and that's called meroblastic. Okay, and then of course holoblastic again. Then these, I will throw at you these terms, isolithical and mesolithical, a little bit later on. But with that slide, I'd like to explain what is holoblastic and meroblastic. Let's go over the first one, spiral cleavage. What happens during spiral cleavage? Here's the spiral cleavage. The first two planes of cleavage, are vertical and produce blastomeres. This is a blastomere, this is a blastomere, this is a blastomere, this is a blastomere. So when they say blastomere, is when you have, this is a zygote, this is 2 cells, 4 cells, 8 cells. So this is a blastomere right here. That is a blastomere right here. So when you have blastomeres of equal size, so the sizes of the blastomere is equal as you go along. And then the third cleavage is horizontal. So what am I saying in this diagram, in the spiral cleavage? The first two, if this is zygote, this is your zygote, the first two cleavage occurs like this, vertically, okay? And then the third one occurs horizontally, like this. And that's spiral cleavage. And I have, did they give you examples? Yes, I gave you some examples. And then the third one, the blastomere are unequal. The blastomeres, they become unequal after the third cleavage. So the first two are vertical, and the third one is horizontal. And then you might say, Amir, this is a round thing, right? This is round. What is vertical, what is horizontal? It goes back. The scientists use the vegetal pole and the animal pole as a point of reference. So the vegetal pole and animal pole determine where is the vertical and horizontal. So I gave you some examples of mollusks. Example of mollusks that they do spiral cleavage. Mollusks, clam, snail. Those are all octopus. Those are all examples of mollusks and annelids, earthworm, leech. Those are all examples of that you are familiar with. You studied them. You've heard about them in your life. Those are examples of annelids. Radial cleavage, on the other hand, right here, what happens with radial cleavage, the first two planes of cleavage are horizontal and produce equal blastomere. The third plane of cleavage is also horizontal. So it's happening, the first two planes are horizontal like this, depending on where in that animal vegetal pole and animal pole is. And the second one is also horizontal, and so on and so forth. And those are, I gave you a few examples, and they're not unequal. Starfish, you already know starfish, and frog. We have model of frog in the lab. And that would be example of radial cleavage. Okay, rotational cleavage, which we human are rotational cleavage. It goes back after the second cleavage, one pair of blastomere comes in a right angle. It means they have, the cleavage occurs after the first few. The cleavage occurs in a right angle. Okay, that's pretty much what we human are, example of rotational cleavage. And here is an example of it, as you can see, right here is in a right angle, the cleavage occurring in a right angle. Discoidal, and what happens discoidal, the cleavage is restricted to a small disc at one end of the embryo, example birds, because they have a huge amount of yolk, so cleavage happens at one end. I don't know, have you opened up any egg in your life and you see a little dots of blood or something? I don't know if you've seen it or not. That's what happens. The rest of the egg is healthy, normal, but at one end of it, the chicken egg that you use, the cleavage is occurring. That's called discoidal. The next one, the other cleavage is superficial in case of arthropods, so it happens in all around right here. So that would be in case of your insects, yolk is in the center, and the cleavage is outer surface, example of the arthropods. So those are the other type of cleavage. Protostome, mouth first. Proto, as you know, based on your prefix and suffixes, it means first, and a stome, stomia, it means mouth. So mouth first, and deuterostome, it means mouth second. Stome, again, deuter, it means second, and mouth second. So types of, I will come back to those. Types of yolk, these are different type of yolk. Isolithical, like starfish, that has equal, iso, it means equal amount of yolk. Okay, mesolithical, it has moderate amount of yolk. Telolithical, it has a good amount of yolk, a lot of yolk. And then centrolithical, it means the yolk is in the center. That's pretty much what those mean. Blastula is a whole mass of cells, and I'm just throwing these terminologies at you, but I'm not, blastocell is the space inside of blastula. Gastrula, having two layers of cells with an opening, and then invagination, archentron. Archentron is embryonic gut, and I will talk about invagination in a minute, hang on. And then coelomic vesicles is formed on the sides of the archentron. Now gametes is an embryo and has 3 germ layers, ectoderm, endoderm, mesoderm. And pretty much ectoderm becomes body surface, epithelial cells, nervous system. Endoderm becomes epithelial cells, GI tract. Mesoderm becomes muscles, reproductive, and diploblastic animals, they have 2 germ layers. Diploblastic animals have ectoderm and endoderm. Triploblastic animals have 3 germ layers, ectoderm, endoderm, mesoderm. Know the fate of blastopore in protostome and gerstome animals. That's something we're going to talk about. Male stages are already, this is in case of human, you guys. Sometimes, some semesters, I did ask, I don't know if I ask you guys, fertilization, of course, we don't talk about it. Zygote, we don't talk about it. Morula is 4 days, and I will talk, what is morula? I will hang on. And then blastocyst, in case of human, they call it blastocyst. The rest of animal kingdom, they call it blastula. Okay, so for human, it's called blastocyst. Gastrula, embryo, and then fetus is after 9 weeks. So those are the time scales. Again, this is for human. You never know, I might ask you these timelines as well. But that's the order. The zygote, morula, blastocyst, gastrula, embryo, and then of course fetus. I'm waiting Or what is this one? What is it supposed to be? Oh, just coming with itself. Oh, okay. Okay. Let's, here this diagram showing you the protostomes and virostome animals. Oh, no! Darn it. Okay, that's okay. Let's go over these models. I gotta turn it on. So let's go over these models. That's talk about these a little bit. What happens, guys? And then I'll go over the PowerPoint 3 minutes. Okay. So what happens when you have this is model of right here. This is model of starfish, and I will go over it in a minute. Am I in there? I hope I'm in there.
I moved it down a little bit.
Okay, that's fine, thank you. So here is the, remember we talked about zygote, and then 2 cells we do not have it. After 2 cells is 4 cells. After 4 cells is 8 cells right here. And then after 8 cells is 16. Then this mass of cells, which is same size, almost same size as zygote. Is that right? Almost same size as zygote. So you have this mass of cells inside of it also is full of cells, like a baseball, like a golf ball. Do you guys follow what I'm talking about? A golf ball, baseball, inside of it is full of material. So that stage is called morula. So after zygote, in the lab, I will not ask you on the microscope, I will not ask you what is this, a 2-cell stage, 4-cell stage. So, but when it gets to 16 or 8 sometimes, this ball of cell is called morula. So definition of morula is when you have a cell after zygote stage, when you have a cell, a bunch of cells that even the inside of the cell is, inside of the structure is full of cells. That's morula. So most likely, this is also a morula. So morula, morula, and then possibly this is morula as well. I don't know. And then the next one, look at it. The inside of the ball of cells, of course the cells, the blastomere are smaller. You guys see that? The blastomeres are smaller because mitosis keep occurring, cleavage keep occurring. So you have morula, and after morula, then you can cut the blastocyst in case of human, or blastula in case of rest of animal kingdom. You cut it, and you see inside of it is hollow. So that stage is called blastocyst. I'm in order. Am I making some sense? This is in order. Blastocyst in case of human. The rest of animal kingdom is called blastula or blastula. Okay? So you have a ball of cells right here. And then what happens after blastula, then the cells, the next stage is gastrula. And I put it up there, you already wrote it down. After blastula, this is frog, or starfish, this is starfish. After blastula is gastrula. Look what happens. The cells on one end, the cells on one end starts folding. It starts, you know, you have, this is a, for example, this is a blastula right here, and all of my arm and my body is cell, 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 and right inside is a fluid filled empty space. Right here is a fluid field. It's not full of air. Okay, it is a fluid field. No cells. Okay, what happens in gastrula, the cells, this is blastula, the cell starts folding inward. And that's called invagination. When the cells starts folding inward, invagination occurs, just like this model. So here are the cells, and the cell starts folding inward. So, some animals, this invagination occurs, this portion right here becomes mouth. And the other end, this invagination occur all the way to the end. Am I making some sense? So this end, mouth, and that end, anus. Right? You have mouth and anus. I have a joke, I'm being recorded, I cannot say it. But in the lab, I'll say it. Hey, what the heck, who's gonna watch this? Anytime you kiss somebody, is that right? Anytime you kiss, you're kissing a tube, half full of poop, is that? Next time we can kiss somebody lovelier, I guess. So try this. This opening right here is mouth, and the other end is anus, and that animal is protostome. Animal, protostome. Proto, I wrote it on the board. Proto, it means first, stome, stomia, mouth. What if you have an animal, like human, we human are like this, same thing. You have flacula, you have gastrula. This becomes anus, and then this becomes first. The invagination occur, remember the invagination is occurring, the invagination is occurring, invagination is occurring. So this first opening, the first opening becomes anus. And then the second opening becomes mouth. Those type of animals are deuterostomes. Do I make sense? So, if I go back to my PowerPoints, which I lost, right here. So, I am making a list of differences between the animals who are protostomes and deuterostomes. Okay, so here is a first cleavage, second cleavage, third cleavage. You have a blastula, you have a blastula. Let's compare ourselves, deuterostome animals. Let's compare ourselves, deuterostome animals with clam. You don't know what clam is? Snail, garden snail. Garden snail, human. Okay, so what happens? You have blastula, same thing. Then you have the invagination, gastrula. You remember that? You have invagination, gastrula, right? But this, in case of protostome animals, that opening, look, it becomes mouth. Forget about the rest. I'll come back to the rest. Okay, this opening right here becomes mouth. And the second opening, the other end, it becomes anus. But look at us, human. We human, that opening becomes our anus, and the other end of tube becomes our mouth. That's the difference between deuterostome and protostome animals. Well, one of the differences. There are a couple more. So, deuterostome animals, most of them undergo, all of them that I know of, undergo radial cleavage. And I gave you the definition of radial cleavage in a minute. And then protostome animals, they undergo spiral cleavage. Most of the animals in clade Lophotrochozoan, that's why I wrote it down here, they are protostomes. And I will talk about clades and all of that in a minute. Not today. We don't have time for it today. Maybe it comes up, I don't know. Okay, so those are the two main differences for now. Now let's look at the rest of the PowerPoints. Are you guys there? Is there any question? For the rest of the semester, I will talk about deuterostome, protostome animals. Quiz on Thursday, deuterostome, protostome animals. Then spiral cleavage, radial cleavage. Okay, so here is the other one, the differences between protostome, deuterostome animals. But I'll show you a better one, a better looking diagram. This is the difference between deuterostome and protostome animals. The deuterostome animals, they undergo intracoelus, and protostome animal undergo schizocilus. Schizo, it means split of coelom. Coelom, it means space. Okay, that's what coelom means. So what happens? You're looking at the animal, right? They cut the animal, embryonic stage of the animal, from here, and you're looking at it from top. Do it make sense? And same as this one. So what happens in case of schizocillus? When you have gut, they show it. I'll show you a better picture. When you have gut, the cells at the corner of the gut, they start splitting. And that's what they call it, schizocillus. And then they form the mesoderm layer. And inside of that mesoderm layer is an empty space which is called coelom. Okay, and all of this will come to a picture a little bit later on, but hang in there. Then, in the intracellus animals, here is your gut. This is the gut. This is the skin. This is the gut. This is the skin. Okay? The gut comes from endoderm, and I will get into that here in a minute. But in the intracellus animals, that outpocketing, there is an outpocketing which becomes coelom. At the end of the day, they are same, right? They're coelomic animals. They have coelom. Okay, so if that picture is very confusing, let's go to this one, which I like it better because it says it all. Here is the schizocillus animals. Here is your blastula. Okay, here is the yellow is endoderm. I will come back to that. Here is a blastocele, right? Empty space, full of fluid. And then the cells here start splitting. And these cells will become mesoderm. Amir, what is mesoderm? Hang on. I wish you would have studied a little bit ahead of the game, then your brain would be more sponge than right now. Okay, so, and then there is space in between in mesoderm, and then at the end you have the same thing as schizocillus animals. What happens in intracellus animals, you have the gut right here. For example, this becomes mouth or anus. Then what happens, they have outpocketing. There is no split of cells. It is outpocketing as the embryo grows, separate from the gut. And then of course you have coelom in here. You have the coelom is surrounded by mesoderm. That's what definition of coelom is. I don't know how much you're familiar with the term coelom. Coelom is a space that is completely surrounded by mesoderm. I mean, what is mesoderm? Hold on to your pants. I'm going to go over that in a minute. Is there any? Yes, sir.
So it won't end up the same? What's the advantage? Like, why do they start with?
Well, scientists, curiosity kills the cat. So scientists starts looking into this. I'm not sure, you're asking me a good question, evolutionary wise. Of course, we have, evolution has answer for everything. But there is an advantage or disadvantage to which way it goes, and I don't know that. But when scientists looked at animals, they saw some animals do it this way, and some animals do it this way. Okay, so they use this criteria. In old days, nowadays everything is based on DNA. They use this criteria to classify animals. So these animals go in this group, classification, taxonomy. So these animals do it this way, they go into this group. That animals do it that way, embryonically. This is all embryology, right? We're talking about how the zygote becomes a starfish eventually, right? Okay, so that's what scientists, they looked into it and they recognized that, the animals who do it this way, they belong to that group, animals do it that way. Human, in general, if you go back, let me go back since you asked that question. Generally, it doesn't. Okay, generally, what happens, animals that are protostome, they are schizocillus. Generally, I said generally. Animals that are durastome, they are intracellus. However, human is durastome, no question asked, but we are schizocillus. That's why I possibly do not have an answer for your question. What is the advantage of being deuterostome and being Schiz cells. We human are like that. We human are deuterostome, but we are Schiz cells. Okay, but I said in animal kingdom, most animals that are protostome, they're Schiz cells. Most animals that are deuterostome, they're intracells. But we human, well, when I say human, most of the primate, mammals, I don't want to go any farther than that, are like that. They're Schiz cells. They're deuterostome, but they're Schiz cells. You see what I'm saying? When I talk about this a little bit later on, what we say, you learn that most primitive animals were evolved first, they're protostome and they're Schiz cells. Animals that evolved later, they are deuterostome animals, like starfish. Starfish is deuterostome. And starfish is also intracells. But we human are deuterostome, but we are Schiz cells. Ask evolution to explain that. Okay, so here it is. Again, this is a nicer diagram of explaining the difference between intracellus and schizocellus. Animals, what happened in animal kingdom, if you have mosaic like mollusks and regulative, most deuterostome animals are like this. So if you have the fertilized zygote and you have 4-cell stage and you separate them, again, generally, you say, oh, we want to do this. No, I don't know. If we separate them, these 4 happens, they can become similar to identical twins. I will talk about that. So if they separate, you separate them in laboratories, they can become a sea urchin, they can become a true organism. But however, in a mosaic model, this is a regulative model, a mosaic model, if you separate them, then some of them become defective. And in case of identical twins, what you have a zygote in the fallopian tube. The zygote under a complete division becomes 2, where the whole set of chromosomes go to 1 cell, whole set of chromosomes go to 1 cell, and become identical twins. Fraternal twin is a different story, but identical twins, and then each